So hello to everyone. I would like to thank to everyone who joined us today for this seminar, especially to Professor Jessica, who kindly accepted our invitation. So just a brief introduction before she starts. Professor Jessica her PhD in biomedical engineering by the University of Texas at Austin and was a postdoctoral fellow at the same institute. And nowadays, she's a senior associate consultant in immunology at Mayo Clinic in Arizona and an assistant professor of immunology at the same institute. So Dr. Lancaster is an expert on T-cell immunology and advanced microscopy techniques. So her group investigates age-associated alterations to the immune system, focusing on the development and response of T cells and B cells by using advanced live cell imaging. So her group directly visualizes T and B cells as they interact within the organ microenvironments in order to understand how communication within the immune system breaks down with age in models of immune response to infection and vaccination, and also cell interactions within solid tumors. So thank you so much, Professor Jessica, for being here with us. The floor is yours now.
Thank you so much for that kind introduction, Luisa. I'm sorry if I keep having to drink water. It's very dry here because we're in the desert. So I'm going to go ahead and get started. Thank you so much for inviting me to your institution to speak virtually. I already feel some affection and closeness because I happen to work alongside many of your former colleagues. And so it is with my pleasure I get to show our lab's research looking at age-related changes to the immune system. So it comes as no surprise that as a society, globally, we are aging. In fact, at least here in the United States, the census has stated that within the next 40 to 50 years, the number of individuals that are over the age of 65 will be roughly the same as those that are of the younger ages. And thus we will have a greater percentage of aged Americans and aged individuals worldwide more than ever before in the history of our planet, likely. However, though changes to medicine and civil infrastructure have greatly increased our life expectancy, unfortunately, this hasn't equated also into an increase in health span being defined as the number of years that an aged individual can expect to live in reasonably good health. As you can see here among several of the countries, especially my home here in the United States, that the average person could expect to spend the last 10 years of their life in rather poor health. And so not only is this tragic on the individual scale to have such a diminished quality of life, but it also creates quite a burden on society as well to be able to care for these individuals, and especially as a large group of these individuals are needed to be cared for by society. And thus, aging is not only an issue for those that are becoming older, it is also an issue for society as a whole. Now, aging science is a rapidly growing field, and it's been very challenging to study because aging is very biologically unique to the individual. In fact, chronological age isn't always the greatest predictor for biological age. And in fact, what biological aging is still doesn't have a ready readout to actually measure it. However, the field of aging research has agreed that there are several major pillars, as we would know, of aging research. I'm just checking the chat to make sure that I'm not missing anything. It looks like it's just people saying hi. There we go.
Hi, Jessica.
Yes.
You don't need to worry about the chat. Luisa and us will
monitor it.
We'll take care of it. And if there's something that come up, we will let you know. Okay.
Oh, please, yeah. Feel free to let me know. So there's several major pillars of aging, and they range on all levels of the biological scale. For example, we have changes to our genetic material and how it is regulated. We have changes to our macromolecules, how they are processed, how they are generated, how they're cleared, especially when they are inflamed or damaged. And then we have changes to the way our cells are able to regenerate, especially to this kind of amount of stem cells that are allowed to replenish all the cells of the body. And then finally, we have the way that all our organ systems are able to adapt to stress as a whole. But the pillar of aging that ties these all together is this kind of reoccurrence of a chronic low-level inflammation. So the circulation of these inflammatory markers and proteins in the blood that seem to change the biological mechanisms of all of the cells of the body. And so the immune system is no different from any of the organ systems in that it is also impacted by aging. And in fact, the immune system is one of the organs that feels the changes to the immune with aging earlier in life. And this has become much more apparent, as I had previously alluded to before the talk, with the advent of the SARS-CoV pandemic, in which the majority of the hospitalization and outcomes for death occur in those that are roughly age 50 or 65 and older. As a system, the changes to the immune system are described rather globally as having a decreased magnitude and delayed response to infectious disease. But at the same time, there is a hypersensitivity of the immune system in which we have increased incidence of autoimmunity, especially among aged females. While it's been appreciated that increases to genetic mutations, that this burden is also creating more chances for cancer as we get older. There's also a big component of the immune system at work here, in which while the younger immune system is readily able to control the advent of tumor cells, unfortunately, as we get older, these immune checks start to decline, and the immune surveillance against tumors goes down, leading to increased likelihood of tumor evasion of the immune system. So, as we said, these broad changes to the immune system have been fairly well described both within biological basic research and also in the clinic. So there are many components of the immune system that decline in their function or their numbers or their composition with age. For example, the naive component, as well as the primary lymphoid organs of the bone marrow and the thymus, change structurally, as well as change in their output of fresh new immune cells to the body. And then we have changes to basic mechanisms such as phagocytosis and antigen presentation by our antigen presenting cells. And then we have decreased production from the humoral response, such as by antibodies and our response or sensitivity to vaccines. At the same time, we have increase in many components that we can consider more inflammatory or can be more aggressive against the self. We have this increased skewing of hematopoiesis towards the myeloid compartment. So we have accumulation of macrophages. Aging in the macrophage compartment is very difficult to study because of the changes to our ability to isolate them or even identify them as we get older. We have increased in many cytokines, especially those that are ascribed to tolerogenic or immunosuppressive, as well as inflammatory functions on both sides of the coin. And then we have an increased accumulation of memory cells, which makes sense because we have a lifetime of pathogen response that is powering the generation of lymphocyte memory. But unfortunately, many of these memory cells have dysfunctional qualities in which they have diminished effector responses and their kinetics of response also decrease. So while we have been able to really define these changes with age, the cellular and molecular mechanisms that are contributing to these changes have been poorly understood. And without this knowledge, it'd be very difficult to create therapies that could possibly rejuvenate or at least improve immune outcomes for the age. So today I would like to talk about two major projects that our lab is studying in the context of the aged immune response. In the first part, I would like to talk about our research, which relates to the decrease in naive T cell numbers as we age. And then the second part, kind of an ongoing research project in which we are trying to understand how aging is leading to changes in the vaccine immune response. Okay, so for the first part, we have published research very recently in which we are trying to understand how aging is leading to changes in T cell homeostasis. So just to kind of remind you, or also as a primer for those who haven't had a formal immunology class yet, we have this pool of T cells, and T cells being white blood cells that are able to target specific pathogens or antigens of those pathogens with a high degree of specificity, allowing them to be cleared out of the body and infect and resolve infection. We have this pool of naive T cells, which are inexperienced against their particular antigen that they're specific for. And they're continually maintained by the body in a homeostatic proliferative process, which allows us to maintain this compartment. And it's very important because these are the cells that allow us to target new and new pathogens that we have yet to encounter. Now, once this naive T cell encounters its specific pathogen, it must be activated, and this is done with help by accessory cells known as antigen-presenting cells, here represented by a dendritic cell. And once this T cell has been activated, it has gone out and has cleared the pathogen threat, it has resolved the infection, and thus part of them will go on to generate a pool of memory T cells, which will allow you to be able to quickly respond once you re-encounter the same pathogen. So as we age, we have a steady decline of naive T cells rather early in life and through midlife, but then their numbers and the proportion within our T cell compartment start to decline rapidly in the latter decades of life. Now, this is also observed in mice. So mice become a very handy organism in which to study this immune decline. We can see that there's a steady decrease in the percentage of naive T cells of both CD4 and CD8 flavors in the blood, but then it starts to decline rapidly at the later months of mouse life. So while typically in aging research, a lot of it is genetic, so people study things like yeasts, tapeworms, and flies. Here in immune aging, we study most of our research, at least in the mouse level, so that we can mirror things that are happening in the human. So I became really interested in this idea of T cell aging because I was wondering how the aged environment itself could be changing the ability of naive T cells to be maintained with age. And this work was kind of inspired by many scientists before me, just like all scientists working today, in which I saw this research in which they adoptively transferred young naive cells into either young or aged hosts. So now these hosts have been sublethally irradiated that would be depleting part of the hematopoietic compartment. And thus, any cells that are adoptively transferred in would be encouraged to homeostatically proliferate in order to rebuild the numbers of T cells in the host. There are mechanisms that maintain a steady number of T cells and B cells in circulation all throughout life. And that seems to not be too perturbed with age. Now, what we saw is that for these young naive T cells, once they were transferred in and they were labeled with a cell proliferation dye, a cell trace violet, we can see that there's many more there's another round of proliferation when we had transferred the young cells into the young host rather than into the aged hosts. And now this was interesting to me because most of aging T cell research focuses on intrinsic changes to T cells. So there are kind of many of the likely suspects, changes to hematopoietic stem cells, telomere shortening, all of these things, indeed, T cells are being susceptible to as we age. But what I had found with this research and what others had done in similar research is that even if you had a young T cell, it was responding differently to its environment, depending on whether it was a young or an aged environment. And so this became very interesting to me. So what is this environment that the T cells are responding to when it's, and how is that different between the young and the aged host? So T cells, like B cells, they're going to circulate through the blood all throughout the body as they conduct immunosurveillance. But to expedite this process, they are going to pass through basically little depots that are strategically positioned throughout the body. And these are the secondary lymphoid organs, which include lymph nodes, as well as the spleen for filtering bloodborne pathogens. And now the lymph nodes are very, they improve the efficiency of the immune response, because now instead of T cells having to go to all the body barrier surfaces, they can just pass through the lymph nodes and thus kind of speed up their ability to scan the entire body for infectious threat. So within the lymph nodes, we have antigen presenting cells such as macrophages, dendritic cells that were scanning the body barrier tissues. They return through the lymph to the lymph nodes, and thus they are able to present any pathogens that they had encountered to the T cells, and the T cells can rapidly scanning them, looking for any possible specificity of their T cell receptor to a possible threat. So it became really interesting to understand how these lymph nodes can be changing with age, since they are so important for coordinating the immune response. Now, if you may realize you all have lymph nodes, and as you have a local infection, these lymph nodes tend to expand in size, and thus the lymph node is structurally made so that it's flexible enough to allow for expansion, which is basically the proliferation of lymphocytes that are specific for a pathogen that's been recognized. However, what has been noticed is that for many aged individuals, this expansion is either incomplete or not to the same extent. And here we see cross sections, H&E sections from the mouse, in which after a young or an aged mouse was immunized, we see that, with the relative scale, the aged mouse's lymph nodes did not expand to the same extent. And indeed, this is often seen in the clinic, in which many aged individuals that have local infections, such as of the throat, may not have present with swelling of the lymph nodes. Thus, there seems to be a difference in how our lymph nodes respond to infection with age. So we used immunofluorescence in our young and aged mouse colony when I first started my laboratory here at Mayo, to kind of try to understand how these lymph nodes can be changing with age, not only with respect to their ability to change in size upon infection. We could see that in the cross-section of the young lymph node that we have this very discrete compartmentalization, where B220 in green is showing us where the B cell follicles are located around the subcapsular zone. And then using a fibroblast marker, PDGFR beta, we see that there is a T cell zone where fibroblasts are holding the T cells here within the paracortex of the lymph node. Unfortunately, when we looked at the aged lymph nodes, we do see that there's a loss of this strict organization between the B cell follicles and the fibroblast network. In fact, we see that the purple for the fibroblast network seems to be everywhere, and we are really having a hard time seeing this very clean delineation of the B cell follicles. We used another fibroblast marker, alpha smooth muscle actin, to see where some of this fibrosis could be occurring with age. So once again, you see that the cross section of the young lymph node, we have this green, which is podoplanin, a marker of a certain fibroblast we will talk about in more detail. It forms a very nice reticular structure, which is important for the contractility of the lymph node during infection. And then we have deposition of alpha smooth muscle actin that's associated with it, another fibroblast marker. This seems to be more centered around the lymphatic junctions and in the medullary sinus, kind of supporting the lymphatics in that way. However, what we see with age is now almost the reverse image, where we have a lot of alpha smooth muscle actin staining here, even into the paracortex where the T cells should be, very similar to what we saw in this PDGFR beta staining. If we were to look closer at higher magnification into this network, and then we can use surface model rendering with the Mayo software, we can see that in green, we have this nice structural network that is forming the scaffold of the lymph node, and then it's dotted with some alpha smooth muscle actin. However, you see here with aged, even though you have some of that green fibroblast network there, it's heavily infiltrated with alpha smooth muscle actin. So you could imagine that this will change some of the physical properties, the mechanical properties of our lymph node if there's a lot of smooth muscle actin deposition in between our fibroblast network. So in my research, I'm very interested in how the immune response, how the components of the immune response work together as a system. And this is because we have the use of a specialized advanced microscopy technique known as two-photon microscopy. So just to kind of give you an overview, two-photon microscopy operates on different physics from what occurs during standard fluorescence. Normally in regular one-photon fluorescence, you have one photon of light that's being absorbed by a fluorescent molecule. And then after a little bit of vibrational decay of energy, you have another photon that is being released of lower energy, and that is what we collect as fluorescent light. However, by two-photon, what we get is a very localized fluorescence response. And this occurs because there are two photons that are absorbed simultaneously in place of that one photon during normal fluorescence. Now, this is a very difficult event to achieve. It requires a very fast pulse of high-powered light. But in doing so, you're able to excite fluorescence in a very fine point. And so you might see here in this cuvette of fluorescein, there's a single dot, and that is where the fluorescence is being excited by two-photon. On the other hand, by regular fluorescence, not only are you exciting the focal point, but you're also exciting the fluorescent molecules above and below this focal point. And so once that's all being collected back into the objective, now you're getting a much more hazy, difficult to resolve image from your thick specimen. Hence, we always look at thin specimens when we're looking with standard fluorescence. But because we have two-photon fluorescence, we're able to look deeper into thicker pieces of tissues, even into intact organs, and we're able to observe them live by looking at it as a time lapse. So we could see here some earlier image I had done. Oops, I fixed it, go forward. Some earlier image I had done in which we can see deep into the mouse thymus. So we have some air positive MTECs that are being surrounded by CD11C positive dendritic cells. And not only can we see them and have a pleasing visual of what's occurring in the thymus, but we could also use software in order to quantify cellular contacts, cell distance. I don't know if I could play this now. And thus obtain quantitative data of what is occurring in real time. So we use this technique to look at the difference between young and aged tissues. And to tell you the truth, as a postdoc, I really struggled with looking at aged tissues. They are quite fragile. And I would tell my supervisor that there was no way I could do it, that the tissues would fall apart. But after a lot of perseverance, it finally worked. So I brought this technique and this knowledge with me to my own position and started using it to look at the lymph nodes versus the thymus, which was my focus during my postdoc. So if we look in the young thymus, you can see here our familiar bean-shaped cross-section here. We see that the capsule is made of collagen, and luckily collagen is a naturally fluorescent material by two-photon fluorescence. It's because of the ordered linear structures formed by the collagen fibers. So those are picked up, as well as a lot of collagen in the sinuses. We had adoptively transferred young naive T cells that were GFP positive because we wanted to see how they migrated in the young versus the aged environment. So as we zoom in, here we go. We could see that these young naive T cells are able to migrate quite freely within the T cell zone. We can use our collagen to see where the boundary is of the lymph node. We know that the B cell follicles are around here somewhere, so there's a little bit of a gap. But then the T cell zone, we see a lot of these T cells that are migrating around quite freely as they're scanning, looking for their specific antigen being presented by dendritic cells. As we get looked into the aged mouse, for one thing, there was a lot of variability in the shape of the lymph nodes. They seem to become a lot more disorganized. And not only that, the positions where we can find T cells was a lot more limited. They seemed to be more confined into even smaller zones of the lymph node. And then there was many collagen-rich areas in which we didn't see any T cells able to migrate in. Now, when we look into regions in order to capture 2-photon videos, we see a much different picture than what we saw in the young case, in which we see that the T cells seem to be trying to crawl along an environment that is full of collagen infiltration. And you can see that when we track these cells, that they have a much more confined, much more limited migration pattern, especially compared to what we see in the aged environment. So we were able to track many of these and use some software in order to model where the collagen is based on the fluorescence signal. And thus we compared the velocity and the path confinement of these T cells when they migrated in the young versus the aged environment. So just keep in mind that these T cells came from the same young mouse. But when we place them in the aged environment, there was a significant difference, a reduction in their mean cell velocity, as well as their ability to travel straight with a greater displacement from their origin during the video. And so we wondered, well, maybe this is just because there's so much more collagen that is available in the aged environment. So of course, as you age, there's more collagen that we're seeing in these lymph nodes. So maybe it's just being close to collagen. And while that explains part of the story, when we sort them into cells that are far away and close to collagen, we see that still there is a shift when you look at young cells that are in the young environment, far from collagen, versus those that are in the aged environment, also farther away from collagen. This was defined as at least one cell length away from collagen, that they still have some sort of dampening in their ability to migrate. And so we wondered that since T cell migration in the lymph nodes is so important for their immunosurveillance and also their function, could this have repercussions on their ability to survive and to activate immunity? So in the 2-photon imaging, this was all done with just collagen being visualized. There's a lot of black space and we don't really know what's there. So we need to kind of understand what is in the T cell environment rather than just other T cells. So the lymph nodes are not just bags of T cells, but they do have a scaffolding that we alluded to that is formed by reticular cells known as fibroblastic reticular cells. They form this kind of organized structure. In fact, they hold the lymph node together. They create this integrity. And then not only are they structurally supporting, holding the lymph node open, but they also form a network for T cells to migrate upon, almost like a highway. And they're able to do that because they have expression of certain survival factors and homeostatic chemokines that allow them to interact with T cells. So not only are the FRCs expressing ligands for the CCR7 receptor, the receptor that naive T cells use to navigate their environment and be recruited to the lymph nodes, but they also express these homeostatic cytokines such as interleukin-7. And this close interaction with FRCs is thus necessary for naive T cells to stay alive and to be maintained within their environment. But as we saw with our kind of gross macroscopic immunofluorescence staining, there's a lot of changes to the to what is in the environment of the FRCs. We see increased deposition of very fibrotic molecules such as alpha smooth muscle actin, as well as collagen. And then we haven't talked too much about this, but there's also deposition of adipocytes. And these are another way that these mesenchymal cells can be altered in their maturation. They could be maturing into adipocytes as well. And this occurs in many of the tissues as we age, not just the lymph nodes. So the fibroblastic reticular cells, these FRC stroma, have been traditionally a little bit more challenging to study. And I feel it's only in the past 5 to 10 years that the field has kind of grown up studying the immunology of stroma and of fibroblasts. It's because our techniques in order to study them have greatly improved. And now we're able to, sort and really study how these FRCs and other stroma change as we age. So they're not a monolith. In fact, they undergo a distinct maturation process of themselves. They start as mesenchymal precursor cells, which become committed to a lineage known as the lymphoid tissue organizer lineage. As they mature, they eventually become precursors to the FRC before they finally mature and become the fibroblasts that maintain the lymph nodes. It is this mature stage that expresses the homeostatic cytokines and chemokines, CCL19, 21, as well as IL-7, which are so important for supporting naive T cell migration and homeostasis. So we did some, qPCR just to kind of understand how some of these markers might be changing with age. We had taken lymph node stroma from young and aged mice, depleted CD45 positive cells, and looked at expression of these homeostatic markers. And while we don't see clear changes to CCL19 or IL-7, we did see a decrease in this basal expression of CCL21. And we, since this chemokine has such importance in T cell trafficking, we wondered if this could be changing some of the things that are occurring in the lymph node. Now, it's been, well appreciated now in this field of stromal biology that these LTOs mature to the FRC terminal state in a lymphotoxin beta dependent manner. So we decided to look at expression of the lymphotoxin beta receptor in the aged stroma. And we did see that there is a significant decrease in the expression of this receptor as we age. But interestingly, though, the expression of the ligand, lymphotoxin alpha, beta, as well as light, which I haven't shown here, doesn't seem to change as we age. We looked at young and aged hematopoietic cells. And so it seems that even though this ligand is available to signal through the lymphotoxin beta receptor, the receptor itself seems to be going down with age. So, when we are beginning the study to look at changes to lymph node stroma as we age, we started with a technique with some quantification that many others in this direct field had looked at. We used flow cytometry in order to determine if there was any quantitative differences to FRCs as we age. So going back to our CD45 depleted stromal cells from the lymph nodes of young and aged mice, we at first didn't really see very much differences in the skin draining lymph node sites. So here we had scrutinized the inguinal, brachial, cervical, and axillary lymph nodes to look at skin draining sites. And you can see here we have FRCs. They don't change in proportion as we age. And neither do these other subsets that are demarcated by podoplanin or CD31. So these are endothelial cells, lymphatic endothelial cells, blood-associated endothelial cells, and what is known as double negative, just because it's negative for both markers, but they seem to be more likely precursors to the FRC compartment based on what's out there in the literature. Interestingly, though, the mesenchymal lymph nodes, so those that are feeding the gut barrier surfaces, they do seem to have a difference in FRCs. So they have a significant decrease in the percentage as well as the numbers of FRC within the mesenchymal compartment, and somewhat of an increase to set that off in the blood and the blood-associated endothelial cells and the double negatives. So it seems at least that the lymph nodes age at different trajectories as, whether they come from the gut and whether they come from the skin draining site. And I think part of that is, our greater appreciation how microbiota can be regulating different biological states. But we wanted to see if, we really wanted to study and understand how FRCs could be changing with age, to go back to our naive T cell question. And so we took our FRC compartment here, and then we further subsetted them based on the upregulation of VCAM. And so this adhesion molecule becomes upregulated as the FRCs become much more mature. So what you can see here in the young compartment is that you have your immature FRCs, you have somewhat of this intermediate stage that we can see and gate out. And then we have this mature stage that is VCAM1 high. But interestingly though, in our aged mice, we see these distinct stages, but you almost see this buildup or accumulation of FRCs that are VCAM1 intermediate. So when we quantify it, we see that there's a decrease in this kind of precursor VCAM1 low stage and an increase in the VCAM1 intermediate. Some of the mice, it looked like there was less VCAM1 high, but we couldn't quite see that significantly based on our mouse numbers. But interestingly, even though skin draining lymph nodes were different in their VCAM1 subsetting, the mesenchymal lymph node cells, we didn't see a difference. So it seems like mesenchymal lymph node cells, less FRCs, but they're all of the different stages. Skin draining, there's the same proportion, but this proportion has been redistributed with age. So we further kind of looked at these subsets based on their expression of lymphotoxin beta receptor. We see that this receptor is especially diminished in the most immature FRC subsets, as we could see here, especially in the skin draining site, though it seems to catch back up by the time we get to the terminal stage. And then it seems to be slightly decreased overall in the mesenchymal compartment. We further looked at this marker of mesenchymal stem cells known as SCA1. It's also in many other cell types. And we see that it's also diminished as we look at the aged, especially the immature FRCs. And so there may be differences in the proliferative potential of these FRCs as well. So just to kind of give you a general model of what we think is happening in the naive T cell compartment as we age, we have this nice lymph node in which we have this network of FRCs, and they are very important because not only do they hold the lymph node open, but they also are covered by IL-7 and homeostatic chemokines, which are very important for naive T cells to traffic along the lymph node as they maintain their homeostatic potential and interact and look for possible pathogens that are being presented. Unfortunately, as we age, there are some changes. We're still kind of testing hypotheses as to what this mechanism could precisely be, but we kind of suspect it has to do with low-level inflammation that occurs with aging. But we have changes to the maturation of the FRC compartment. Now we have an increased accumulation of these more immature pro-fibrotic FRCs that are receiving less lymphotoxin beta signaling because their receptor levels have changed, and also their cell cycle could be changing as well. And now our T cells are encountering this very different environment in which they're not able to access those few remaining mature cells that do present their survival cytokines, and thus we are having this decrease of naive T cells. And this change could also be further impacting the FRCs in an almost feed-forward loop, because now we have decreased naive T cell maintenance. They also express factors like lymphotoxin alpha and beta that are important for maintaining the stromal compartment, and thus you're having this kind of avalanche or runaway effect of lymph node decline as we age. So with that, I would like to kind of, we're continuing work to really understand why lymphotoxin beta signaling could be impacted, what could be changing the FRCs as we age. But I kind of want to talk a little bit about another story that's in progress in the lab, just to shift gears a little bit, looking at how these changes to the T cell maintenance could be impacting the humoral response. So just to kind of show what our motivation is here, we have started to understand that the elderly have decreased responses to many of the vaccines that are available. And that's generally because vaccines are developed in younger animals and humans. It's very expensive to be able to study aged animals and to to have aged cohorts in which to study vaccines, though it is becoming much more common as we appreciate these changes. But it has been noted that many vaccines are less effective in aged individuals. And currently the strategies are just to increase the antigen dose for the most part. That's why you hear about different flu vaccines available for the aged. Not only is antigen dose being modulated, but often we have changes in the adjuvant to try to make them a little bit more immunogenic. So for example, if you have the seasonal flu vaccine, it's up to 90% efficacious in younger adults, but about half the effectiveness into older adults. So even though they have these alternate vaccine regimes that could be, increased in dose to be possibly boosted up, they're seeing that the long-term readouts are not as impacted. The generation of memory isn't as good. So it seems that if you vaccinate younger, it seems to have a better preservation of memory responses than if you are getting the same vaccine as you get older. So why is this important? We need to understand why aged immune responses are changing so that vaccine biologists can optimize their designs against many of these vaccines that are particularly important for safeguarding older populations against RSV, also for bacteria that are more virulent against aged individuals, as well as when we start to design more vaccines for targeting cancer. So just to kind of remind you of the basic biology that's being perturbed, we have our naive B cell, and it's recognizing its antigen through its B cell receptor. It becomes activated. And at the same time, we have naive CD4 helper T cells, also recognizing antigens from the same pathogen that are being presented by the dendritic cell. They become activated, and then they are going to come together into a TCR MHC interaction event. And it is during this prolonged interaction that the CD4 T cell, now activated, is going to further stimulate this B cell and allow it to be terminally differentiated. And it is this activation that is necessary in order to have some of these long-term, very helpful benefits of the humoral response, such as the generation of plasmablasts that are generating high affinity antibodies, as well as the generation of memory B cells that are able to respond quickly when we re-encounter a pathogen. But what is occurring with age? So many studies have described there is a significant reduction in the outputs that we see from the humoral response. We see that there's lower affinity antibodies being generated, much fewer plasmablasts, as well as reduced memory B cells. And so since we know that this interaction event is so important, I became curious how this could be changed within age, especially knowing that aging is changing the tissue environment that these lymphocytes interact with. So, the initiation of this response has been well studied, especially by kind of pioneers in two-photon fluorescence. For example, in the group of Jason's sister from UC San Francisco, they were able to kind of very elegantly show that upon activation, the CD4 T cell and B cell are going to travel to the boundary within the lymph node where these two compartments reside. It is there where they're going to form a long-lasting interaction, but it is something that is known as an entanglement. It is a long-lasting interaction that is also motile, very different than what we see with T cells that are arresting during antigen-specific activation. And in fact, it's almost this prolonged dance that we see in which the T cell is dragging its B cell partner around as it's providing its activating cytokines. And so, in my research, we are very sensitive to the idea that this interaction event occurs within intact tissues, and that being within this 3D architecture can really change what could be occurring, and it'd be very different from what you can see, for example, when the cells are simply together in an in vitro environment. So in the lymph node, just to remind you, it is organized into distinct compartments where we have our B cell follicles, and then we have our T cells in the T cell zone. And these two compartments are both supported by different subsets of fibrotic stroma. We have our FRCs that we already talked about that are in the T cell zone here in orange. And then we have what is known as the follicular dendritic cell, which is not a dendritic cell like the APCs, but also just a type of stroma that seems to have that shape. And it resides in the B cells to support the B cell follicle. So within these organized compartments, we have this formation of what is known as the germinal center. So the germinal center is where these highly activated, terminally differentiated B cells are able to differentiate into their plasma cells that will be generating the antibody. And so, you know, this interaction and this germinal center initiation and maintenance has been, you know, very carefully studied, at least for young organisms, in which we see that these B cells, once they've been activated by the T cells, will enter the germinal center and begin the cycling event between the dark zone and the light zone in order to be selected for the highest affinity B cells that can go on to mature and become our long-lived plasma cells. So we're interested in turning our two-photon technique to really understand kind of this very elegant, very dynamically coordinated response that is going to be occurring within the germinal centers of the lymph node. So we are currently aging out this FRC specific promoter. It's a CCL19 EYFP mouse, in which we can see that we have some, you know, based on the brightnesses here, we can see that there's a lot of these T cells in the T cell zone, the T cells being labeled in red. And then the B cell follicle, since we're not labeling B cells here just yet, you know, it's kind of empty because most of the T cells are not quite trafficking in there. But there's this very nice boundary zone between the T cells and the B cells, in which we have these FRCs that are kind of coordinating the crawling of the T cells towards the follicle. And you can see here when we zoom in that indeed, using our FRC specific promoter, that we see these T cells very actively crawling and jumping along this FRC network as they navigate in the lymph node. So, you know, once again, kind of calling back to Jason's sister's work, who has done so much work studying the FRCs and of the lymph node, it's starting to become more appreciated that there is a lot of heterogeneity within the FRC compartment, and that this can be important for promoting the immune response. So you can see here, we have part of our cross-section, B cell follicle again, T cell zone here, but now we have different subsets of FRCs, especially these T cell zone FRCs that are lining the interfollicular regions between the follicles and between the T cell and B cell zone. Not only that, these stroma are able to support the migration of the two different types of lymphocytes, the T cells and the B cells, based on their expression of the cytokines CCL19 and CCL21 in the T cell zone, as well as CXCL13 in the B cell zone. And so work from other groups has shown that upon immunization, that the old mouse here, the stroma from the old mouse, is not able to upregulate the expression of some of these homeostatic cytokines upon infection. Here we see that there's a diminished activation of CXCL13 as well as CCL21. And so this can really impact the ability of the T cells and B cells to respond to their environment. Namely because the CD4 T cells upregulate CXCR5 so they can move towards CXCL13, while the activated B cells upregulate CCR7 so they can move towards CCL19 and CCL21. We could see here, just like in our imaging, that these FRCs are very important for T cells to crawl towards the follicle region. Here shown in blue, we see with ERTR7, this very nice red network of FRCs extending even into the B cell follicle. And not only that, Ann Haberman's group in Yale had done some really nice imaging where they see it's actually this interfollicular region at the boundaries of the follicle that allow for the T cells and B cells to make contact with each other. So there's something very special about this niche because it has a distribution of chemokines that allow for both of these cells to come together once they are activated, whereas otherwise they would be so segregated into their distinct compartments when they're in their naive state. In fact, the FRCs, even though they're associated with the T cell zone, are very important for the continuation of the immune response. Here we see from Shannon Turley's lab that, their research does a lot of work with stroma immunology. They found that if they ablated the FRC compartment, that, these once nicely formed germinal centers are now very poor and kind of all over the place. We have B cell staining everywhere. And we also lose the ability to form flu-specific antibodies in their immunization models. So these FRCs are just very important for trafficking the lymphocytes into their correct position in order to allow for the germinal center response to occur and be maintained. Not only do lymphocytes expand upon immunization, but these subsets of these FRCs, their numbers also expand. So there's proliferation that must occur within the FRC compartment to support the expansion of the lymph node upon immunization. And we could see here, if you look at the little white square compared to the circle, and then the white triangle down here, especially in the mesenterics, we saw a diminishment in the expansion of the FRCs, and that the proliferative potential of these FRCs were also changed as well. And so there seems to be changes until not only at the FRCs at homeostasis, like in our earlier research, but also in the way that they respond to immunization. If you have a question, you can always ask again. If not, I'll finish. I'm almost done. I know we have just a couple minutes, sorry. So, I wanted to understand how these germinal centers are changing with age. My background had always been at T cell homeostasis, thymic development, and so immunizations were very new to me. We started with this peptide immunization to get ourselves started. So we immunized young and aged mice with MP ova. This is nitrophenolhaptin and ovalbumin in alum adjuvant. And we did confirm that, at least for the MP specific B cells, that we have this robust generation of the germinal center B cell phenotype in the young mice, but that this is greatly diminished in the older mouse. But that could be a problem of aged T cells and B cells. So we really wanted to see what could be the contribution of the aged environment itself. So we took young T cells and young B cells that are specific for the MP and the ova, and we placed them, we adoptively transferred them into mice, which were subsequently immunized. These are young and aged mice. And we did find that there was a decrease in the expansion of the germinal center B cells specific for MP, looking here at the aged, but it did seem to be highly variable. Some mice do expand, but it wasn't as consistent as with younger mice. But the most striking difference that we saw was in the T follicular helper cell phenotype. Looking at these OT2s here, we see that in the aged mice, we have expansion of CXCR5 positive PD1 positive OT2 cells. So we have a robust expansion of the T follicular helper phenotype upon immunization, but with aging, we didn't see this quite as dramatic change in the expansion of antigen-specific T follicular helper cells, whose important role it is to support the germinal center response. So current work right now, it's very time-consuming and tedious, but we're trying to see what this looks like as we age. Our current research right now, we've been doing imaging in the young mice just to kind of determine what is the timeline of immune activation and germinal center initiation in response to our peptide model. So we can see here in our naive mouse, we have plentiful antigen-specific CD4 T cells that are migrating around quite rapidly, and we have some of these B cells that migrate a little bit more slowly. But then one day after immunization, all the T cells just stop in their place. And not only that, they are clustered, presumably around antigen-presenting dendritic cells, and thus they're, undergoing motility arrest. As we go to the next day after that, however, while we see some of the cells that are still kind of clustered and moving very slowly in regions where we probably have dendritic cells holding them there with antigen, we see that a subset of these T cells are now activated enough towards the T follicular helper phenotype, and they're moving to what is probably the boundary with the B cell. And not only that, now we have activated B cells that are starting to make probing interactions with our T cells. And, for this was very striking and exciting for me to see. We always think of a circle meeting a circle and then they're being activated, but you can see that the B cells are almost very amoeboid, as that they're probing and trying to make activation with the T cells. And then if we go a couple more days, we see that the expansion of these lymphocytes are quite complete. Now everyone's moving quite rapidly through the environment, and we have this formation of a spectacular germinal center B cell cluster within the 5 days. And so our goal in subsequent imaging is to do this over the progression of the days and really see what could be the difference. I'm having a hard time finding that. Okay. So, what could be going wrong during aging? So this is, being able to see this will really inform our future investigations as to what we will want to look at. Could it be the initial TCR signaling with the dendritic cell, or is it with the TCR signaling that's occurring with the B cell? That will form new avenues of research for us to look at. Will we see differences in the motion speed of the T cells? We already feel that there may be differences in the chemokine distribution. The increase in fibrosis can be changing how quickly T cells can move towards the follicle. And then finally, what could be different about the conjugation event itself? I don't have time to show it, but we are seeing differences in CD40 ligand upregulation with age, but there could be differences to co-stimulation, SLAM family members, other things that are there to support the T cell and B cell contact. So our goal is to use imaging to kind of track how that could be changing. And I'd like to just kind of quickly summarize. With aging, we do see that there's changes to the architecture of the lymph nodes being, important centers for the immune response to be coordinated. We see age-related changes to the FRCs. Their differentiation may have repercussions on how lymphocytes are being maintained and activated. And then we are doing some continuing work now to see if this disorganization may be impairing the T cell dependent activation of B cells. And then finally, if we can restore aged tissues, could we possibly restore the immune response? And I'll kind of skip these questions because I'm sorry I've gone over time, but thank you very much for your attention. I'd like to thank the hardworking members of my lab. Also, we have close ties with our neighbor lab, the Borges de Silva lab, who some of you may know. And then we have some collaborators that are helping us in ongoing work. Thank you very much, and I'll take any questions.
Thank you so much, Dr. Jessica. Amazing presentation and very beautiful images you have.
Oh, thank you so
Fantastic.
much.
So we are open to questions right now. If you have questions, you can just open your mic and raise your hands or put it in the chat.
Yes.
Hi, Luisa.
So,
I have a question.
yes, of course, Hugo.
Hi, Jessica. Nice talk.
Hi, thank you.
I would like to know if you checked the phenotype of T cells comparing aging and young mice using activation marks or looking for the profile of the cells.
I haven't done it personally, but many groups have. So, especially, I look more at like CD4 T cells, but both compartments do change in their effector functions. So there's decreased expression of certain effector cytokines. TCR strength of signaling seems to be changed. Even like the ability for the cytoskeleton to polarize to kind of get TCR elements into place. So there are definitely T cell intrinsic changes. So we're trying to uncouple that from what could be just the aged tissue itself.
Yeah, I think this cell is more exhausted, maybe.
Yes, definitely. That's why we're transferring like the young cells in to kind of see how they're responding. But yeah, definitely there's a lot more exhausted T cells, PD-1 high T cells as we age.
Yeah, thank you.
You're welcome. I see there's Tiago.
Yes, Thiago.
Hello. I have a question, but you can actually take the questions from the students first. It's usually how it goes on the seminars. I can ask after the students.
Yes, of course. So we can go to Rio de Janeiro. I think the audience have questions.
Yes. Okay.
Thank you. Good afternoon. My name is Lucas. I'm a student here at the Federal University of Rio de Janeiro, and I have a question. Thank you, Professor, for your presentation. It was really amazing. Your work really caught my attention because it's really consonant, similar, not similar, but it runs side by side with the work of Cecilia Cavazzoni. It's a colleague of ours. She was interested in how the differences between an age group and a young group to SARS-CoV vaccines, and how the immune response in these mice occurred. And she got to a really amazing conclusion in which she observed differences in follicular T cells of the regulatory subtype, in which they increased in aged group, but in comparison with the young group, and the follicular helper cells had a decrease in the age group, and she correlated this with the more deficient immune response and humoral response in the age group. Have you thought about inquiring about the role of the regulatory and helper follicular cells in the lymph nodes, because they are present also in the lymph nodes? That's my question.
Yes, I'm definitely fascinated. I'll have to look at her research as well. Yeah, because I know that there are the follicular regulatory subset, and that it seems that in her research, I think there are others research as well that seem to suggest that this subset is getting expanded. And that kind of really mirrors what we see with like Tregs. Tregs are also found to be somewhat expanded as we age. And I think, this is such an important question because, immunosuppression is increasing with age. So the T cells can be dampening themselves or, diverted towards that phenotype. And so they can't carry out their regular follicular helper function. I think I need a collaborator or the bandwidth to be able to help me. Right now we are focused on the stroma. That's where the imaging is kind of taking us, but I know that there's so much more to the question of how T cells are changing with age. So I definitely wish I could. I think we need more hands to help. More experts. Mm-hmm.
Thank you so much.
You're welcome. Great question.
We have more questions from Rio de Janeiro.
I do see that there's a question.
Yes.
Oh, there's a question.
But I'm waiting for the students first. Yeah. Maybe they have.
No. The students here are done for now.
Okay.
I have a question, but I'll ask whenever my time comes. All right. Thank you.
Okay. Jessica, thank you for the inspiring talk. Really, it was a very stimulating opportunity to hear what you presented to us. I have a very specific question, which is related to one aspect of your story that you almost didn't touch, which has to do with the high endothelial venules and the endothelial cells. Because as you mentioned, the environment, based on your experiments, transfer experiments of young T cells to an old mice, the environment is clearly, shaping the outcome. Of course, it could happen already within the lymph node in the stroma itself. But I mean, if you really take into consideration the fact that, for example, cardiovascular functions are so much influenced by the endothelial impairment of endothelial functions and alterations that happen as a function of the inflammatory status that we have during aging. Why should not expect that the high endothelial venules are also reflecting that transition? Because it could either influence the stroma, so you could, imagine the interplay happening with the stroma. It could influence the selection of the T cell subsets that are entering the stroma. There are so many interesting questions regarding to the potential role of endothelial cells. And I remember that I asked Michel Nussensweig when he first came to Brazil and presented his marvelous data with the multiphoton microscopy that we only saw that for the first time when he came here. And I asked him, because we were always working on some aspects of this story, and I asked him, what do you know about the permeability of the high endothelial cells? Because if you have increased permeability, you should have actually extravasation of plasma components into the stroma. So maybe you could have interesting possibilities there. Can you expand on that, or do you think that I'm too far away from what your present thoughts on this?
No, it's quite fortunate that you ask about that. I regret that for me to limit myself to the FRC, the fibroblast stroma, was just kind of like a practical decision, but I definitely recognize that the lymphatics and the endothelial cells are playing a tremendous role in some of the differences that I'm saying. For example, Mike Diamond's imaging research that he did, he had West Nile virus infection, and he studied the diapedesis of the T cells into young and aged organs, and found that there was decreased recruitment. Our adoptive transfer also suggests that I'm just seeing decreased recruitment into the lymph node that could be explaining some of the differences that I'm seeing. And there is some very nice electron microscopy publication out there where they looked at the structure of the endothelial vessels, and they do see that there is more leakiness, more permeability that could be really allowing certain proteins in, and also kind of changing the way that cells are being recruited. So I think part of my challenge now, especially looking at this Germinal Center project, is how do I uncouple my adoptively transferred lymphocytes? Their failure is partly due to failure of recruitment and retention. How do I uncouple that from their failure to be in the environment? Yes, it's something I wish I could study more. We just, as a new lab, we don't have quite the bandwidth. And so I was kind of forced to make a decision to exclude them. But yes, all the data shows, that we've seen that there are changes to them, and that's impacting aging.
Well, that's very, very, very interesting. I will send you a brief message just to collect the references and maybe to exchange some ideas with you.
Yes, definitely.
Thank you so much.
Yeah, thank you, Julio.
Before we go to Thiago's question, we have a student question here in the chat from Caio Loureiro. He asks, hi, Dr. Jessica, congratulations for the presentation. Have you assessed the other markers of immunosenescence on T cells in mice besides PD-1, markers like KLR-G1, although it does not have well-defined immunosenescence markers for senescence in mice?
Yeah, I mean, I haven't looked so much at immunosenescence of T cells. We did look at immunosenescence of the stroma, and at least in the aged mice that we looked at, we didn't see, we did SA beta gal staining, and we didn't really see any staining quite there. We did see some changes to the stroma. We had, with P21 gene expression, also inflammatory cytokines like IL-6 were highly expressed. But for the T cells, I don't know, I feel as, I have the great fortune to be working within the same state as Dr. Janko Nikolic-Zugic, who is an expert in T cell aging. He's done some looking at KLRG1 and things like that in his research. So I'm starting to work more with him. I'm hoping he can kind of have the manpower to kind of look at the T cell response while we're looking at the fibroblasts. So even though I'm like a T cell biologist at heart, I feel like the way I can be more impactful as a new investigator is to kind of really understand this aging stroma and then how it supports T cell differences. So unfortunately, we haven't looked as much changes in T cells other than reading out, PD1 and, CXCR5. Though interestingly, I think some of my students' research looking at PD1 seems that the upregulation is not quite so complete when these young T cells are put in the aged mice to start the aged T follicular helper response. And so we're starting to wonder if that could be important. So, but wonderful question. Yes, all these great questions. Everyone has these wonderful ideas. I wish I had all the time, multiple copies of me and, multiple postdocs to help, because there's so many wonderful things to look at. Thank you.
Tiago?
Yes, thanks for waiting.
Hi, Jessica. That was a very nice presentation, very nice talk. Thank you. My question is regarding the differences that you observe in the lymph node architecture. So in the skin or in the lungs, let's say, or any other tissue in the body, usually collagen deposition, it's related to a wound healing process, right? There's tissue damage, and then lots of collagen gets accumulated there. So I was curious about how, the mechanism behind this accumulation of collagen and actin, also actin that you observe in the draining lymph nodes of aged mice. Do you have any insights related to that?
Yeah, so in our continuing research, we really want to understand what could be driving this kind of accumulation of fibrosis and collagen. And then certainly kind of the usual suspect is, macrophages undergoing wound healing response, if they're becoming more inflammatory or more kind of turns towards the wound, I guess not quite inflammatory, but if the inflammatory of the conditions of the aged environment are kind of turning them towards more M2, allowing them to kind of be stimulating collagen deposition. I feel like that is the obvious answer, but I think that because I see changes to lymphotoxin beta receptor, I still feel like there's a problem of stroma differentiation. But yeah, that's going to be the challenge, is how to uncouple, because undoubtedly there's some sort of aberrant wound healing response occurring. It happens in other systems, other aged tissues. So we haven't looked too much because, I just know that the system of looking at these wound healing macrophages and other cells could be kind of complex as well. So, but it's definitely the first thing that we'll probably have to rule out if we're going to come up with any mechanism for what we think is occurring. So,
okay.
Unfortunately, I guess my short answer is I haven't looked at it yet, but I would love to. I know it's there. Yeah.
Sounds good.
Thank you. Should I pick someone, or do you want, Lisa, if you want to pick?
I think Miriam is the next. Miriam?
Actually, Miriam gave me her turn because she's away from the computer. She's moving near it. But meanwhile, I'm going to ask my question is, what role, if any, natural selection plays in influencing aging-associated changes? or if these changes that we are seeing are just a consequence of a process that is actually influenced by natural selection. And specifically is the amount of immunological stimuli that these animals see during their lifetime may influence how much collagen deposition and how much the stroma is changed with aging.
Okay, so it seems that there's two distinct questions, I guess. So
well, hopefully they will connect somehow. That's
yeah.
They're connecting in my head, so it's I may have to further explain how I'm getting there, but please.
Okay, well, I guess as far as the first question, I mean, why would our immune systems decline with age? I mean, it's interesting also because there are mammals, like marine mammals, the naked mole rat, that don't have these, you know, alterations. You know, they don't decline as much in their immune systems or their anti-cancer responses as they age, while we do, and the mouse and non-human primates do. I mean, I guess if I were to suspect, and some people have said this, the evolutionary pressure is that as we get older, we need to make time for the reproductively fit members of our species. So they will eventually outcompete, you know, the older, you know, so I guess, so reproductive ability and immunosenescence seem to kind of be opposite ends of the spectrum. As we are the most reproductively fit, we are the most immunologically healthy. But as we lose that capacity, then our immune senescence can start to set in. And I think part of that is maybe there's hormones that kind of dictate it. But I think those are all theories that I don't know enough about. So you wanted to know how this kind of ties with fibrosis.
And this can be some sort of collateral effect of the response that these animals are seeing throughout their lives. And also connecting with Thiago's question, that maybe these inflammatory environments that these lymph nodes are exposed to, these can be a reflection of it. And if so, can it be actually worsened by exposure to immunological stimuli throughout life, or it does not change? It doesn't matter if the mice or the man are exposed to different infections while they're young, and on how they will actually age later on, or the lymph node will age later on. Does it matter how much exposure to immunological stimuli they have?
Yeah, I'm not quite sure. I mean, as far as like why our lymphoid organs would be degraded, I think it's like a question of probably energetics. Like all our tissues start to become more frail because I think as you know, as we have evolved so that we don't really need to maintain older, unfortunately, older individuals to continue the species. And so like, you know, as we get older, maybe we don't eat as much, we don't expend as much energy. And so I think also the tissues kind of reflect that, you know, perhaps there isn't quite the energy to kind of maintain this fully differentiated state. And so kind of fibrosis and these other mechanisms start to set in. But as far as, I think probably acute, so I mean that this goes to like hygiene theory or things like, I think maybe acute exposures that we can fight are probably good for the immune system. But I think aging kind of mirrors like chronic infections that we never resolve. And so there are like studies where if someone has chronic HSV or something, and then they give them an acute exposure of another, to me, it looks a lot like what I see with the aging phenotype, with like the diminished response, the diminished, you know, immune outputs. So I don't know. I mean, it might be too controversial to say that, yes, having, you know, consistent infections throughout life makes you healthier. I mean, because there are people that are more susceptible to infections and they don't seem like they're necessarily living a higher level of life, and there could be differences. Because I've noticed people who are born prematurely, just so anecdotally, I've noticed that some of these individuals tend to get, acute infections a lot more with a lot more severe magnitude. So they'll get colds that are more persistent, or mono, infections that are more devastating. So even though they consistently get little acute infections, I don't know if they're any healthier, but I haven't done any study on that, so I can't back that up. It's just something I've noticed. So
thank you. Midian has a question.
Yes.
Thank you very much.
Yeah, you're welcome. It's a philosophical question, I felt worthy of much discussion.
Thank you very much, Jessica. And that's the last time I let Bruno ask before me, because he kind of took my question away.
Sorry.
I'm joking, but I was going to ask if you had tried making multiple immunizations to the mice and seeing it, and looking if you will expedite the accumulation of collagen, or if you make it slower to happen, or if you saw genetic change, if you tried using different mouse strains, and if you saw also a different speed as the tissue get replaced and you get more and more of the accumulation of the collagen over there. So I guess you kind of already asked, but I'm not sure if you're planning on doing any of that later on. And if you compared and saw more of the CD844, CD8 positive, CD44 double positive cells, when you see a decrease in the naive cells, if you were counterbalanced by an increased population of antigen experienced of those virtual memory cells, or any sort of other CD8 T cell population in these lymph nodes.
Okay, so first, the multiple immunizations I haven't really tried. So, I must admit I'm like very inexperienced to immunizing mice. So even just this MP-OVA challenge was something new for me to learn and have my staff do. As far as like kind of perturbing fibrosis, like we do have the CCL19 pre-lymphotoxin beta receptor floxed mouse that we're trying to do some studies on. In this mouse, the adult lymph nodes are prematurely fibrotic. So we kind of want to study how T cell motility, the immunization response kind of works in this mouse system where the lymph nodes are fibrotic even in the young mouse. But we haven't played with the immunization yet to see if that changes fibrosis. I have to admit, even though like the fibrosis looks kind of striking by immunofluorescence, it could be variable. Like some mice don't look as fibrotic as we age. And I think it speaks to the variability, even in this Black 6 mouse, like aging impacts them differently. And then I've also kind of talked with pathologists here at Mayo and asked, like, in human lymph nodes, do you notice fibrosis? And they don't really see it as this overwhelming thing, not like in cancer or in HIV. Like they agree that there is fibrosis there, but for them, it's not as detectable by eye in the human. So even though I found studies that say like there are fibrosis by certain markers, it's not as overwhelming. So like some of the things that fibrosis is doing, I think it's still kind of subtle. I mean, obviously, as we get older, you don't have no immune system, like you just have a slightly dampened ones, which is why I still feel it's an important question to look at fibrosis, even though it's not like as aggressive as a tumor fibrosis. And
because it might have a bias depending on the background of the subjects, right? If you were in a more sterile condition where you have some vaccines where you stimulate more often versus a person who deals more with the earth and is more exposed, maybe you might have a different evolution besides the genetic background, as you mentioned, that you might have differences in humans. And so I was wondering if you could mimic that either with the obesity, or do you see any differences between male and females that could have the hormone maybe giving? It's such an interesting topic that we come with all these side questions besides what he has shown, but your work is really amazing. So it's just thinking about all the possibilities actually. And
yeah, we haven't seen quite any like strict differences in the fibrosis. I have to admit, like the aged mice, you know, they're quite costly, so I can't be as like I can't do as much as I want to really determine, but at least like, you know, by flow, by immunofluorescence, I haven't seen anything as like pops out. So obviously that looks different, but maybe like if I were to do a much more careful study, we could see that. We have noticed that when we do these MP-OVA immunizations, that sometimes, you know, we seem to have a more robust response in the females. So we do have to kind of repeat that a little bit with our males versus females groups, because we just kind of want to check that before we actually publish something with all of them lumped together. So right now we're doing some repeats to kind of see. But I was surprised that the female was such a robust response, but to see.
Okay, thank you very much.
You're welcome.
Would you mind if I just jump in? I just wanted to follow what Megan was saying, is that although, I mean, the immunization protocol, I think is a very nice idea, because one of my hypotheses, although you seem to believe that might be something related to macrophages, which could be as well. So I actually wondered if this collagen deposition might be something related to a reprogramming of those T cells, or perhaps even macrophages. There has been some cases in the literature showing that repeated immunizations lead to a trained immunity. So although the most obvious thing would be, you know, like tissue damage in the lymph nodes, it might be also something related to reprogramming of those lymphocytes or
yeah, that's definitely a good point, because I can recall reading about, because a lot of my fibrosis in the lymph node, I'd read a lot of HIV papers because lymph node fibrosis is something that is observed there. And I think I saw this one paper where they thought it was CD8 T cells that were inducing, you know, then the laying of collagen. And so there's definitely a lymphocyte component that could be explored there, and that maybe just blaming macrophages is the easy answer, but you know, if this could be something quite different from the normal healing response. Yeah. Yes.
Gustavo?
Okay, Jessica, super thanks again for such a nice presentation, and the area that you are studying is quite interesting. So I wish you lots of success.
Oh, I appreciate it.
Area. I think I'm gonna touch back these questions that we are, you know, was first raised by Bruno, then me and Thiago a little bit, and adding something else, that is the plasticity of the system. So first question is how plastic you think are those modifications, and how much these are related to the T cell compartment? Because we know that if you look at the germ-free mice, for instance, or the nude mice or ragnogaults, the architecture of the lymph nodes are different from the regular mice. So I wonder a couple of things. If you have done, or you are interested in doing, the age of lymph nodes in germ-free or in just the nude mice. I think the T cell compartment plays a fundamental role in the architecture. And I know that I remember, you know, work from Ken Shortman, Ron Scola in the WEHI in the thymus. So if you get to mice and give the immune cells, the young immune cells back to those mice, the architecture of the thymus get kind of restored. Mm-hmm. In your case, I think the lymph nodes, once they get fibrotic, so they may be like, you know, there's no going back from that. But if you keep feeding young cells to a point where there's no fibrosis, it's getting aged, but there's no fibrosis, can you delay the fibrosis by giving back the immune cells? So you are looking and you're showing results. In other words, a lot of how the aged lymph nodes, the liver, or is not an optimal place for T cells activation, B cell activation. But what about other way around? The signals from the immune cells to the stromal environment of the lymph.
Yeah, so I mean, you're kind of touching a lot on like things that I'm proposing to do in my NIH application. So I'm hoping that it gets funded so I could do some of these things. I mean, some of the experiments that we want to do is, first, like in vitro, we want to see if we could make a young stromal cell old. So like how, if they're in an inflammatory cytokine environment, kind of similar to what people have done with lung fibroblasts in idiopathic pulmonary fibrosis. They found they could induce fibrosis in those mesenchymal precursor cells and make them a fibrotic lung cell. So I'm wondering, one, can I make a young FRC old prematurely in vitro? And then to kind of reverse it, there are kind of systemic antifibrotic drugs that people use for kidney or lung fibrosis. And so I'm kind of wondering if if it's just a problem of fibrosis and T cells can't move around, they can't crosstalk with the stroma as well, if we can kind of could these therapies potentially reduce fibrosis? Could now signaling occur to restore the lymph node? So those are definitely things I want to try, because for me, I have like an engineering training, so it's not just enough to understand what is happening. I do want to kind of figure out can we perturb or manipulate the system to kind of restore some function. So you're definitely speaking about my 5-year plan, how do we make the fibrosis or make the aged lymph node phenotype occur, and then how can we reduce it? Looking at the lymphotoxin beta receptor, I know that there are agonists that we can stimulate that pathway. If we just really stimulate it, could we kind of restore the environment? Or if we block some TNF receptor engagement, could we allow can we minimize this kind of fibrosis that we see? So many things that I want to try. Thank you. It gives me great comfort to know that I'm trying to steer towards things that other investigators would kind of, instantly identify just based on looking at the work. So thank you. I appreciate that.
Thank you and good luck.
Thanks.
I think we have time for one more question. We have a question in Rio de Janeiro. Cristiane.
Hi.
Hello.
I'm sorry, it's already late, and I would like to ask you a very simple, basic question after all this deep discussion. But I was really curious, and I would like to hear what you think about it. I thought your model very elegant concerning transferring the young cells to the aged mice, so you solve the problem with the intrinsic T cell modifications that age. But I was wondering, as the transference is intravenously, and of course there is already very well established modifications in serum, plasma levels of several factors. So I was wondering, and do you think that during the presence of your transferred cells in the blood compartment, before they reach the lymph node, they can, change there before, of course, your 2-photon images and data concerning the disorganization of the lymph node are beautiful, and the barriers that the collagen triggers, so they block the ability of the T cells to move. But I was wondering if the T cells that were exposed to blood of young versus aged mice arrived in the lymph node differently, with differences.
Yeah. I mean, so I do worry about that, because, like, we are transferring them, and these cells, they've lived in, one blood environment, and now they're experiencing a new environment. And then, yeah, could they be? So I think my only response, and I'll have to think on it and also consult with people who may also have better ideas. I may transfer both young and old cells together, and then hopefully, we can kind of suss, if the aged cells also respond a certain, I don't know, like, or if we took some of the mouse's own cells, or as to kind of see, like, oh,
how long the cells stays in blood compartment before they reach the lymph node after you transfer them? I don't know. I was wondering.
I mean, it's actually pretty quick. I mean, I give them a day just to try to have enough circulation, but I mean, I think I could probably transfer them, and you could start detecting them within hours, or because as the lymphocytes do spend a majority of their time crawling around in the lymph nodes. I think when people have done those experiments, it's like they only spend like minutes between lymph nodes, but then they spend like hours in the lymph nodes. So
yeah, that's what I was expecting to hear. I know I don't work with that at all.
Oh, yeah.
I have no experience with this.
No, I have to trust what's out there. Sometimes you can't do all the experiments yourself, but I think, yeah, it's like they can move pretty quickly into the lymph node as long as they have those signals to recruit them.
Okay, thank you.
You're welcome.
Beautiful work. Thank you.
Oh, I appreciate it. Thank you.
So I think we don't have more questions for now. And I would like to thank you, Professor Jessica, for being here with us today.
Oh, thank you.
It was an amazing talk.
And I appreciated the questions. It was really helpful to me. Yeah.
Thank you so much. And just a message for you all, in the next week, and the same time, we will have the lecture of Professor Hai Jung Kim from Harvard, that will talk about allele-dependent Treg stability in health and disease. So I wait for you in the same time. Okay. Thank you so much, Professor Jessica, for you all.
Thank you, Jessica. Thank you so much. Say hi to Henrique for us.
Yes,
please.
I will. He's right next door. All right.
Thank you.